All right, I just finished class. We ran out of time because I just made a lot of stuff for us to do and didn't realize that, it was going to take so long. But I hope you enjoy this. I posted the breakout room number 2 for you so you could look at the questions, and now we're going to go over the answers. Okay, first question. Which of the following would cause a decrease in skin pigment? And so we have several answers here. And so I will go over that. That must be the people who responded the last time I did this. And so the correct answer is a decrease in tyrosinase enzymes. And that's actually can happen in certain clinical conditions in certain areas of the body, and also on the body completely if you have a defect in creating tyrosinase. And this is actually a histopathology slide of albinism. So if somebody doesn't make any pigment, they don't have any melanocytes to make pigment, or the process of making the pigment is defective, then you're not going to end up with pigment in the skin. And certainly that pigment is there for a reason, for UV protection of the DNA. And so that could cause an increase of skin cancers, which you'll go over when you do clinical applications tomorrow. So that's what this picture is showing. And this enzyme is important for the production of melanin. And that's why a deficiency in it is going to cause that kind of lack of pigment. Okay, the next question. Which of the following would you expect to be most prevalent on the surface of a melanocyte? So, think of a melanocyte, what a melanocyte does. Here are your options: gap junctions, hemidesmosomes, tight junctions, microvilli, cilia. You have to know what these things are to be able to think about a melanocyte and then decide what they might have. And so, if this is what my last class put on there, so the correct answer is a hemidesmosome. So a melanocyte is attached to the basement membrane. It has hemidesmosomes, but they're not attached to the other keratinocytes by any desmosomes or tight junctions. There's no microvilli. They do have processes, but they don't have microvilli or cilia. They just have cytoplasmic processes. And then, a lot of cells have maybe a cell junction, a gap junction here or there, but certainly not very prevalent on a melanocyte. All right, so a hemidesmosome is the correct answer. Okay, so here is actually a picture of a young child with albinism. And so if you have a deficiency in making tyrosinase enzyme, or in making melanin, like you might just not make a whole lot of melanin just overall, then you can have a partial loss of melanin or pigment in the body, or you can have it, just generally systemically, which we get in individuals with albinism. And so I wanted to add this clinical correlation in with a picture, and so that makes it a little bit more connected to melanocytes. All right, so which of the following sensations are most likely to stimulate a ruffini corpuscle? So remember ruffini corpuscles, what might stimulate their firing here? And the answer is stretch or torque. So when I think of ruffini, I put on my socks, I'm stretching the skin, and that's because they have collagen fibers that stick through the capsule. So when you pull on collagen, you're going to stimulate a ruffini. And that's why that happens. Like 78% of my last class got that correct. So which of the following cells or structures do not play any role in sensory reception? A lot of these cells do and structures do, but which one of these do not? So hopefully you put D, a Langerhans cell, because like Dexter, that's what I remember for a Langerhans cell. These cells are looking around for viruses, bacteria, other antigens, so they can mount an immune response and tackle them and kill them and get rid of them. So Langerhans cell is an immune cell. Which of the following layers of the skin would be most likely to be the origin of skin cancers? Let's see what the students put. They put A, the stratum basale, and that's great because that's the right answer. Now, certainly you have a little bit of mitosis and spinosum, but the amount of mitosis in the stratum basale is much higher. And so anytime you have mitosis happening, you can have a abnormal division of the cells and replication of the DNA. And so that mutation can actually go out of control and not checked, and you can end up with a tumor. And so the basal cell tumors would be certainly the most prevalent. So identify the region indicated. So here you're looking at a histology slide, and you have a lot of regions that you're trying to decide which one this is. And most people put D, the papillary dermis, and that would be correct. So here I can see the epidermis. I can see cornified cells, but it's a very light staining slide. You know, I don't like the variation in the slides, but I want to show it to you so you can help look at them and figure out what is what. And so here you can see cornified cells or ghosts of cornified cells. This is the basal layer and the spinosal layer and the granulosome layer here. And then underneath this lighter color, not as deep staining, because here you can see deep collagen fibers, lots of stained collagen, and elastic fibers stained pinkish, orange, red in an HME as well. So these are collagen and elastic fibers. And this is a lot lighter colored. Less density in the fibers and more cellular, but you can see some spatterings of cell. So I would have put papillary dermis as well. All right, so let's look at this area. Here we have the epidermis, we have papillary dermis sticking up. The reason why this looks so funky is because we're cutting off tips of papillary dermis. This slide is cut kind of obliquely, and that's why it looks the way it does. It's not that this slide has papillary dermis within the epidermis, it's that the slide is cut in an oblique angle. And here you have the cornified keratinocyte. So these are the keratinized part of the epithelium, and so this would be the stratum corneum. Okay, looks like other people put spinosum last time, but I don't know why. All right, so let's identify this function of these cells that are outlined. So this is sort of a fun game I like to play, because I'm really asking you to think about where the cells are located at and how do they appear. So these cells are in the basal region, and I drew cytoplasmic processes for you. You can see the cytoplasm is more pale, and there's not as many of them. All of these other cells look very, very similar to each other, and then all of a sudden you get this special cell. Well, let me think of some cells that might be down there. A Merkel cell might be down there, and a melanocyte might be down there. And so those are the two I have to choose from. So I drew these cytoplasmic processes, and that is definitely not going to be indicative of a Merkel cell, because that's really just a small rounded cell. So I was kind of giving you a hint with drawing this pretend cellular processes. So really these are melanocytes, and you don't have to find a melanocyte, but I think it helps conceptually to understand where they're located at, what they do, what they look like, so you can think of them with a personality. All right, identify the structure indicated by this arrow. Here you can see these strangely structured, they look like cells, some nuclei, but it's not really organized like a regular cell. I mean, it certainly doesn't look as neat as these other ones, and that's because this is an encapsulated sensory receptor. And what I'm looking at, big slide, I like to show you big and then magnified. I see cornified epithelium here, and I see non-keratinized down here. So this is the epidermis, awesome, and this looks like a dermal papilla sticking up in the epidermis. Well, what sensory receptor is located in the dermal papilla? Well, that's going to be a Meissner's corpuscle, and that's going to be stimulated by light touch. And so Meissner corpuscle is the correct answer. Okay, what do you think A is in the lumen of, and what do you think B is? B is an advanced question. So A is in the lumen of a gland. I know that because these are kind of cuboidal shaped cells, and they look like they are bordering each other. It could be a duct, but at this point, in looking at my choices here, I got a lot of glands. I don't have any ducts. It's certainly not a hair follicle because it doesn't look like stacked, stratified keratinocytes that are going to become cornified. They look like they're secretory products in these cells as well. And it's pretty large lumen. So I would go with an apocrine gland for this A. Then for B, these are very stretched out long cells. I see a nuclei around here every once in a while. And this in this wall of this gland. That's really interesting. Well, this is actually a myoepithelial cell. So I glossed over myoepithelial cells when I talked about them being a part of a gland. And here I'm showing you, here's the nucleus here, and its cytoplasm is, you can pretend in your mind's eye that it's going to wrapping around here. So these contractile cells help with gland secretion. And so I would have put D, apocrine gland for A, and a myoepithelial cell for B. These dark cell would be cuboid in shape. Myofibroblast. Well, a fibroblast would be in, I would expect that to be more in connective tissue. So this doesn't look like it's in the right area to be a myofibroblast. And so it doesn't look like a muscle that would be around a sebaceous gland. So that's what I would go with. Okay. A. So what in the world could A be? And what is B? So I'm looking at connective tissue, fibers, and all these organization, irregular, dense connective tissue. I'm sorry, I'm losing my marbles. Dense, irregular connective tissue. And then I see rounded structures, some of which might be nerve axons, some of which are glands. Some, I got blood vessels around here too. And so I know you haven't done all of these structures yet, but I still want to start getting you to recognize the patterns. And so I'm thinking that A, because it's pretty round in shape, and this looks like these cells are not cuboidal, and there's lots of them, and they look relatively organized. They just don't look cuboidal at all. And so I'm going to go with a blood vessel being A. It also has, I'm going to call this smuts in the middle. So it doesn't look like a whole lot from this magnification. So it's got some proteinaceous material in here, probably from the fixation process. And it has some cells. These might be white blood cells that are in the middle. And these might be remnants of red blood cells. So I'm going to go with a blood vessel because a nerve wouldn't have this lumen and stuff inside. And a Paxinian corpuscle, which looks like a big onion, doesn't also have a lumen. A hair follicle, there's no hair, and it's very eosinophilic in the wall, and hair follicles have a lot more hematoxylin, or more blue, in the matrix of the hair follicle. And B, this looks like, these look like eccrine sweat glands. These look like ducts here. They're cuboid. This is a nice cross section. There's a lumen inside, and these look like the secretory portions, which are more pale staining. So I'm going to go with a eccrine gland for B and a blood vessel for A, and that's and for those reasons. Okay. So a really cool correlation. This is ink that is deposited in the dermis of the skin of a tattoo. I just really like this when I found this, and I had to add this in there. So there you go. All right. So this slide is the pathologic slide. It's a type of skin cancer. But just thinking about where these cells are, and maybe a little bit about what they look like. What cells do you think this might have originated from? And let's look at my choices. I got squamous cells from the stratum corneum. These cells look like little round cells, and they're definitely not up here in the corneum, so I'm not going to cross that one out. Keratinocytes from the stratum spinosum. Well, these are really purple in color. In stratum spinosum here, these are lighter pink, big fluffy cells. They don't look like keratinocytes. They don't look like they're filling with keratin. Langerhans cells from the stratum spinosum. Well, Langerhans cells aren't going to divide. They're going to be originating from monocytes. And so I wouldn't expect them to become cancers, firstly. Definitely not, if there's bone marrow cancer, they're not going to just pop up as Langerhans cells. And they're also not disrupting the stratum spinosum, so I'm going to cross that one out. Last one, Merkel cells from the basement membrane. Well, you got it. Little small ovoid cells down here close to the basal region of the epidermis, and they're very hematoxylin staining, very blue. So I'm going to go with D for this question. Okay, so that's the answers for breakout room number 2. I hope this helps you. If you have any questions, just let me know.